Ever wonder how doctors figure out exactly which bacteria is causing an infection?
Yeah, it's like a microbial game of Clue, isn't it?
It really is. But today, we're going to dive deep into a test that cracks the case. And get this, it uses a simple color change.
That's right, the Urea Hydrolysis Test.
Urea Hydrolysis Test.
Sounds complicated.
The name's a mouthful, but it all comes down to whether a bacteria can produce a specific enzyme.
Urease.
Okay, hold on. Enzymes, bacteria. You're losing me.
Right, so Urease. It has this thing for urea.
Rhea, like what's in pee.
Exactly. It's a waste product our bodies get rid of in urine.
Okay, so urease and urea, what's the connection?
And Urease breaks it down
Breaks it down, like into what?
into ammonia and carbon dioxide.
So urease is like this little chemical machine that transforms urea.
That's a great way to think about it. And it's the ammonia that really shakes things up.
How so?
Ammonia is a base. It makes its surroundings more alkaline,
Alkaline way.
the opposite of acidic. Think baking soda versus lemon juice.
Got it. And some bacteria actually prefer things a bit more on the alkaline side.
Exactly. They thrive in those conditions.
So they're creating their own little comfy environment.
Like tiny little pH adjusters. But how do we actually observe this in the lab? Well, that's where things get really visual.
Oh, I like where this is going. Tell me more.
Imagine a broth, a special bacterial soup, if you will, that starts out yellowish-orange.
Okay, I'm picturing it.
We add the bacteria we want to test to this broth.
Like adding ingredients to our soup.
Exactly. Now, this broth, it contains something called phenol red.
Phenol red. What's that?
It's a pH indicator. It's sensitive to changes in pH.
So it's like a pH litmus test.
Precisely. If our bacteria are producing urease, they'll break down the urea in the broth, releasing that ammonia, and guess what?
The broth changes color.
You got it. It transforms from yellowish-orange to a vibrant fuchsia pink.
Wow, that's pretty dramatic.
It's a clear sign that urease is present and active.
So we're essentially looking for a color-changing magic trick to tell us about these bacteria.
It's like a visual alert system. It's incredible how such a simple change can reveal so much about what's happening at a microscopic level.
It really is. But this color change, it's not just a cool visual, right? What does it actually mean for, say, identifying bacteria and treating infections?
You're right, it has real-world implications.
Like what? Give me an example.
Take Proteus mirabilis, for instance.
Proteus mirabilis. Doesn't ring a bell.
It's a common culprit behind those pesky urinary tract infections, UTIs.
Oh, UTIs. Those are no fun.
And guess what? Proteus mirabilis is a champion urease producer.
So if we pop a sample from a patient with a suspected UTI into the special broth and it turns bright pink,
Bingo. It's a strong indicator that Proteus mirabilis might be the troublemaker.
it's like a bacterial fingerprint. That's amazing.
Now, on the other hand, let's talk about E. coli.
E. coli, I've heard of that one. Isn't that also a common cause of UTIs?
It is. But here's the thing, E. coli, it doesn't produce urease.
So even though they can both cause similar problems, this test helps differentiate between them.
Absolutely. And that's crucial for doctors.
Because knowing which bacteria is the culprit helps them choose the most effective treatment, right?
Precisely. It all comes down to understanding how these tiny organisms behave and what makes them tick.
So we've got this powerful color-changing test, but paint me a picture. What does this actually look like in the lab?
Well, not as dramatic as you might think.
No bubbling beakers or anything.
This test is all about simplicity. We're basically just setting the stage for the bacteria and watching for that color change.
So no high-tech equipment needed.
We do use something called a urea broth, or sometimes a urea slant.
Urea broth, urea slant. Those sound kind of fancy.
They're just specialized test tubes, really, designed specifically for this type of testing.
Okay, so we've got our test tubes, they've got this broth in them, and the pH indicator too, right?
That's right. And then we carefully introduce a sample of the bacteria we want to test into the broth.
So it's like adding a starter to a sourdough bread.
Exactly. We give the bacteria a chance to interact with their environment, and we see what they do.
And then we just sit back and wait for the color to change.
Exactly. We usually give the bacteria 24 to 48 hours to incubate in the broth.
Giving them plenty of time to do their thing.
That's the idea. If they're the urease-producing type, they'll break down the urea, the ammonia is released, and boom, that color change happens.
And if the broth stays that same yellowish orange,
Then we know those bacteria aren't producing urease, no color change, no urease activity. It's pretty straightforward.
so elegant. Just a simple visual cue to answer a pretty complex question.
It is. And to make things even easier, there's usually a chart in the lab that summarizes the key points of the urea hydrolysis test.
Oh, so it's like a cheat sheet for bacterial behavior.
You could say that. It helps make sure everyone interpreting the results is on the same page.
It's amazing to me how much we can learn from something as simple as a color-changing broth. It's like these bacteria are sending us secret messages.
It really highlights how even the smallest details can reveal a lot about the world around us.
This urea hydrolysis test is clearly a handy tool for microbiologists.
Right, but it's not just about what happens in the lab.
So how does this actually impact, say, someone who's not a scientist?
Let's say you're a doctor and a patient comes in. They've got all the signs of a UTI. Yeah.
Painful, frequent urination, the whole 9 yards.
And you need to figure out the best course of treatment fast.
So knowing whether the bacteria causing the problem is a Urease producer or not, that's vital info.
Exactly. Some antibiotics work better on certain types of bacteria than others.
So this simple test can actually lead to faster recovery for a patient.
That's the goal. It's about getting people the right treatment as quickly as possible.
It's amazing how something that seems so small, this color change in a test tube, can have such a big impact.
And it doesn't stop there. Think bigger picture for a second.
Hey, I'm intrigued. Go on.
Imagine using this test to analyze, say, water samples.
Water samples. What would we be looking for?
The presence of certain bacteria, of course. It could be a tool for public health, helping to prevent outbreaks.
So we're talking about taking this from the lab and applying it to the real world in a really impactful way.
Exactly. It really shows how seemingly isolated scientific discoveries can have these incredible ripple effects.
It's all connected. This has been fascinating, really makes you appreciate the power of observation and how something so simple can unlock a deeper understanding of the world around us.
It's a big world out there, full of amazing things waiting to be discovered.
And we'll be back next time to dive into another one. Thanks for joining us on this deep dive. Until next time, stay curious.